Hello, everyone, and welcome to today's webinar, Next Generation Protein Sequencing, Interrogating Proteins with Single Molecule Amino Acid Resolution on Platinum. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Quantum-Si. To learn more, visit www.quantum-si.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use this to submit any technical issues here as well if you're having trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Ben Moree, Principal Scientist, Applications Development at Quantum-Si. Dr. Moree, you may now begin your presentation.
Hello, my name is Ben Moree, and I'm a Principal Scientist at Quantum-Si. I'd like to thank the organizers today for giving me the opportunity to present my talk titled Next Generation Protein Sequencing, Interrogating Proteins with Single Molecule Amino Acid Resolution on Platinum. So in my talk today, I'm going to discuss three main topics. I'll begin by telling you about how next generation protein sequencing can provide novel insights into proteomic workflows. From there, I'll tell you how platinum can generate single molecule amino acid resolution during protein sequencing experiments. And then finally, I'll discuss several application areas of this technology, including protein identification, peptide barcoding, and the detection of amino acid variants. So with that, I'd like to tell you why I believe platinum should be considered a standard part of your proteomic workflows. Really, what we're looking for in today's proteomic experiments is the ability to have a deeper understanding of proteins and associated biology. We know that proteins are the key determinants for human health and disease, and as such, they're responsible for nearly every action within the cell. However, proteins drive biology. However, to better understand how they do that, we need to understand proteins not just at the primary sequence level, but also all of their associated proteoforms. Currently, estimates of approximately 20,000 genes at the primary sequence level within the genome. However, the entire expressed proteome consists of over potentially 1 million different proteoforms. We therefore need new technologies to help us start to decipher this complexity within the proteome. This is where platinum can potentially step in and provide those novel insights. The way platinum performs its next generation protein sequencing, it has the ability to uncover novel protein variants, proteoforms, isoforms, and potential post-translational modifications, all with single molecule amino acid level resolution. This can lead to deeper insights to help provide a new understanding of how proteins drive biology. With that, I'd like to take a few minutes and describe to you how platinum generates its single molecule amino acid level resolution sequencing. To begin with, I'm just going to give you an overview of the platinum workflow. It consists of 3 simple steps in an easy to use end-to-end solution. Begin by preparing and ready to sequence protein libraries with our standard protein reagent kits. Those ready to sequence protein libraries are placed onto a semiconductor chip, which is then loaded into the platinum benchtop instrument where we generate high precision kinetic data. That kinetic data is then transferred to the cloud where the data is automatically analyzed without the need for any additional bioinformatics expertise, and it's generated reports that are easy to read and interpret. So how do we generate single molecule amino acid level resolution data on platinum? We start by taking a protein of interest and digesting that protein with the proteinase lyc to generate peptides. These peptides are functionalized via the C-terminus with macromolecular linkers that enable these peptide complexes to be loaded onto our semiconductor chip by a biotin strip-avidin interaction. Once these peptides are loaded onto the chip, we add a sequencing solution to the chip that contains our fluorescently labeled N-terminal amino acid recognizers, as well as our aminopeptidases. The N-terminal amino acid recognizers will bind to and associate with specific amino acids through a series of on-off binding events, generate a fluorescent signal that is captured and collected by the chip. This fluorescent signal contains information such as the fluorescence lifetime and intensity information, and is what enables the discrimination of the recognizers onto the chip. During the course of these binding events, we generate a series of high precision kinetic data that enables each individual recognizer to potentially uncover or unlock each amino acid or multiple amino acids within the overall sequence space, as shown in this figure, in which we have discrimination of both the leucine and isoleucine amino acids within the same recognizer. This process continues until an aminopeptidase comes in and removes the N-terminal amino acid from the peptide chain, exposing the next amino acid in that peptide to the sequencing solution. This entire process continues over multiple rounds of sequencing and cleavage of the N-terminal amino acid until the entire peptide chain is finally sequenced. Once this sequencing run is complete, the data is transferred to the cloud, where our bioinformatics platform translates the on-chip kinetic data into the corresponding amino acids within the sequencing run. So how do we take and analyze the data to determine the amino acid cause? We do this by looking at a combination of the on-chip kinetic data. We begin by taking and identifying within any specific kinetic trace a recognition segment. Highlighted above in the figure are examples of 4 distinct recognition segments. A recognition segment is a region within any kinetic trace in which there is a consistent fluorescence lifetime and intensity information, which is representative of these repeated on-off binding events of our recognizers. Once we have identified a recognition segment, we then determine the number of pulses and the average length of each binding event within that pulse, and that value is reported out as our pulse duration. It is these differences in pulse duration within a single recognizer that enable us to discriminate multiple amino acids. As shown again in this example above, we have this leucine recognition segment, which has these longer pulses and an average pulse duration of approximately 11 seconds, compared to the isoleucine using the same recognizer, but with much shorter pulses that average approximately 0.33 seconds. So it's a combination of these recognition segments and the associated pulse durations that enable us to classify and identify amino acids. Once we've done this on a chip-by-chip basis for the entire chip, we can then take all of these kinetic traces and use this information to identify the proteins and peptides within our sequencing sample. An example of this is shown again to the left. On the left-hand side, we have our kinetic traces, which indicate the recognition segments and their associated order of appearance of those segments, as well as the corresponding pulse durations. We then take these kinetic traces that we observe on chip and we compare them to the predicted values from a reference peptide or protein that is associated into our database. This reference information contains the predicted data that includes information about the recognition segments, their expected order of appearance, as well as their pulse durations. We then use computational and statistical parameters to determine the alignment score of the traces relative to any of the particular potential reference peptides within the overall sequence space. Only peptides that achieve a minimum threshold score are considered to be aligned to a particular peptide of interest, as shown here on the far right-hand side, in which we've taken the initial kinetic traces observed on chip and aligned them to the reference traces based on the on-chip kinetics that are expected. Once we have done this at the entire chip level, we've taken all these kinetic traces and we've aligned them to particular peptides within that reference database. We can then use this information to take and determine the data at the peptide level, protein level, and at the individual amino acid level, as shown in the figures above. In this upper figure, what I'm showing you are the total number of alignments for every protein within that mixture. This happens to be a 5-protein mixture in which you can see the identification of all the total alignments to each of the 5 proteins within the sequence. You can then click into and go into a deeper level of understanding and look at the number of alignments for each of the individual peptides found within that protein of interest. And then finally, ultimately, we get at the highest resolution data, which is that of the peptide level, in which you can see 3 different pieces of information displayed. One is the total number of alignments for that peptide. Then we have these coverage maps, which represent, based on the height of the fill within each one of these boxes, the relative frequency of that specific amino acid being identified within the total number of alignments. So in this example shown here, this initial phenylalanine is found in all 100% of these 1,302 alignments, compared to the tyrosine at the end of the sequence, which is only found in, say, 5 to 10% of the 1,300 alignments. Now, in addition to the coverage map, you also get a determination of the global pulse duration values for all of the 1,300 alignments demonstrated here. So we're showing again for all the phenylalanines across all the kinetic traces that align to this peptide, we're reporting out this global value. And it's really this information at this amino acid level that you can use to help determine changes in protein structure that may be associated with such things as post-translational modifications or changes in the protein sequence through mutations by looking at changes in the patterns of either the coverage maps or the underlying post-duration kinetics. So with that understanding of how single molecule sequencing works on platinum, I'd like to now take you into a few use cases of this technology, starting with protein identification. All right, in this example, I'm starting off with a simple protein identification of the protein PD-L1, also known as programmed death ligand 1, which is a major component in the immune checkpoint pathways and is a target for immunotherapies. On the left-hand side, what I'm showing you is data indicating 5 distinct kinetic signatures that were identified belonging to peptides for PD-L1. The peptides are highlighted in the upper right-hand figure with the black boxes indicating the sequence space within each of the peptides that these protein or peptides map back to. Now, this is a use case of a single protein using our standard 5 recognizer mix, which includes recognizers that contain the ability to recognize aromatic residues, hydrophobic residues, arginine, alanine, serine, or asparagine and glutamine. This is using a standard recognition kit, and we were able to identify 8 out of the potential 14 peptides within PD-L1 upon a lyc digestion. Obviously, there are more complex scenarios that we can use platinum sequencing for. And on this particular slide, I'm showing you is data indicating the identification of multiple proteins from a protein mixture. Again, on the left-hand side, what I'm going to show you are the 5 distinct proteins that went into this mixture. This is a mixture containing, again, the protein PD-L1, which I just highlighted, as well as human serum albumin, interleukin-4, interleukin-6, and the protein CDNF, or cerebral dopamine neurotrophic factor. All of these proteins were mixed at an equimolar ratio into a single tube and then digested with lyc, went through our standard library preparation process to functionalize them, and then were immobilized on chip and sequenced on platinum. On the right-hand side, you can see an example of the total number of alignments that were achieved for each one of these proteins in the sequencing reaction, as well as example kinetic signatures of a single peptide from each one of these proteins. Again, similar to the data that I've previously shown, you can see the coverage map for each particular for a specific peptide from each protein, indicating the number and frequency of the amino acid calls within that peptide, as well as the associated pulse-duration kinetics for each one of those particular calls. Now, it's important to note with platinum sequencing that you do not necessarily anticipate that all proteins will generate the same number of alignments due to differences in the amino acid coverage for each particular peptide that may be present within the sequence. I also want to highlight another example in which we can use protein identification with platinum. And this is the case of particular in-gel digestion procedure. In this particular situation, we were using recombinant protein that we ran on a gel, and we found this unexpected slower migrating band highlighted in band B. We were very curious about why this additional band was present on the gel. We wanted to know if we could identify it. So to do so, what we did was we cut out these bands from this SDS page gel. We then performed an in-gel digestion in which those gel slices were digested in the presence of lyc to digest the intact protein into its peptide fragments. Those peptide fragments were then isolated from that solution and subject to our standard library preparation protocol. As you can see in this particular sample, we were able to, for this protein CDNF, we were able to identify using, again, our standard 5 binder recognition kit. We were able to identify 5 distinct peptides from CDNF, each with their own distinct recognition pattern and on-chip kinetics for both the band A and the band B sample. the total number of alignments scaled in a titration and concentration dependent manner, as shown in the graph below. So this is a great use case in which you may have an unexpected band on the gel that may allow this technology to come in and identify an unknown band without the need for any extensive workflows. Now, our normal library preparation process is similar to, I would say, some traditional mass spectrometry workflows in which you take an intact protein, you reduce and alkylate those proteins to remove any potential disulfide bonds. Then you use a protease, such as lysine, to digest these into peptide fragments. These peptide fragments, in our particular case for our library preparation, are then functionalized to produce a C-terminal azide group, which we then use click chemistry to conjugate to our macromolecular linkers. Now, this workflow is a very straightforward workflow. However, we have been able to adapt this workflow for more specific application-based needs, such as the one that I will describe here with immunoprecipitation. And now, immunoprecipitation is a very common technique in biology that's used to isolate proteins from complex mixtures, or commonly also used to perform antibody quality controls in which you want to verify that your antibody is interacting with your target of interest. In the particular case, for us, we had a need to develop an alteration to this workflow because of the harsh elution conditions that typically occur in immunoprecipitation, leading to some incompatibilities with the downstream chemistries. In order to do this, what we developed was this on-bead digestion method, in which we took and performed our immunoprecipitation using some magnetic beads, as well as the antibody-antigen complex. We digested the entire sample to produce, with our lysine enzyme, to produce our peptides, and then essentially continued through our standard library prep procedure in which we functionalize those peptides, click them to our linkers, and then perform sequencing. An example of what this data looks like is shown here above. In this particular case, we performed an immunoprecipitation of the protein interleukin-6, or IL-6, which is a key immune modulator. In this particular experiment, we began by taking recombinant IL-6 and we spiked it into human serum at the amounts indicated in the bottom of this figure shown here. We then took the human serum, depleted it of abundant proteins such as human serum albumin and IgGs, and then again pre-cleared that serum with a control IgG to remove any nonspecific background binding bands. The proteins were then immunoprecipitated from the human serum using a high affinity anti-IL-6 antibody that was coupled to streptavidin-coated dynabeads. Finally, we proceeded with our on-bead digestion process and then continued into sequencing. What you can see in the data shown here above, again, using our standard 5 recognizer kit, we were able to identify 3 unique peptides that align to these sequences from IL-6, as shown above, each with their unique kinetic trace, along with their corresponding coverage maps and pulse durations. So again, just highlighting the ability of this technology to work in workflows such as immunoprecipitation. Now, we've also recently begun looking at the ability for this the immunoprecipitation procedure to identify endogenous proteins. And one such example is shown here above with the protein vimentin. In this particular case, we were able to take an IP from a T75 flask of confluent HeLa cells, vimentin protein, which again, we performed our on-bead digestion process with the immunoprecipitated samples. Shown in the upper left-hand portion is the sequence for vimentin, and in the black boxes are a number of highlighted the individual peptides that we were able to align to from our sequencing data. Shown on the right-hand side are representative kinetic traces of each of those individual peptides. Again, this just highlights the ability of the Platinum technology to take from a complex sample such as a cell lysate and performing an IP and identifying proteins of interest. And we believe this is certainly going to be a very useful work case to extend this into more complex samples and co-IPs. And with that now, I'd also like to tell you a little about another application on Platinum, which is that of peptide barcoding. So peptide barcoding is a process that is analogous to DNA barcoding, in which you can take and essentially tag mixtures of samples to identify them later through downstream screening processes. In the specific use of peptide barcodes for platinum, what we do is we take and append a small peptide barcode at the C terminus of a protein of interest. We can then express and purify those proteins of interest in a mixture of proteins, as shown here in the schematic. Those proteins can then undergo some level of selection, and then after selection, we can cleave these peptide barcodes from that protein of interest, load those barcodes onto a chip and sequence to identify them. Now, this particular workflow is going to be, I think, a very helpful workflow in cases in which your typical DNA-based selection methods that have a genotype-phenotype linkage can be disrupted. This is a very common process that may happen when you're working in host systems that enable secretion. And so with the peptide barcoding, you can continue to tag the identification to the full-length protein and preserve that genotype-phenotype linkage throughout the screening and selection process. So on this next slide, I'm giving you an overview of essentially how this process works, as well as how we've constructed these particular constructs. In the sample above, essentially the workflow for this peptide barcoding uses expression and purification of a barcoded protein. Typically, this is done by including an affinity tag at the N-terminus of the protein of interest, as shown here in the schematic. We then take, at the C-terminus of the peptide barcode, we include an enzymatic functionalization step that introduces an azide group that enables us to perform our downstream click chemistries. Finally, you can undergo your rounds of selection of the protein of interest, and then you can remove the peptide barcode from that protein of interest by the addition of a protease, which is encoded in between the protein of interest and the peptide barcode in the overall protein sequence. Finally, these peptide barcodes can be sequenced directly on platinum once immobilized. So in the example above, what I'm demonstrating here in this slide is a number of different peptide barcodes that we have established for use on platinum. As you can see, all of the associated peptide barcodes in this 8 barcode mixture are relatively small, ranging from 6 amino acids in length to approximately 10 amino acids. What's important for these peptide barcodes and what enables them to work for the unique identification of the proteins of interest is that they all have these very distinct pattern of recognition segments, as well as the on-chip kinetics. And so it's really taking advantage of the power of the single molecule amino acid resolution of platinum to uniquely identify these small peptides based on their distinct on-chip kinetic properties. So I want to give 2 examples of how this can potentially be used in a real-world application. So the first example is a very straightforward situation in which we've taken a mixture of these barcodes. All 8 of these barcodes were individually prepared and pulled into a combined sample. The ratio of the barcodes in the sample are shown here on the left-hand side of the figure. As you can see, the ratio of the least abundant barcode to the most abundant barcode is approximately 10-fold. We then took this mixture of each of the barcodes and performed our standard platinum sequencing based on each of these on this complete barcode mixture. And as you can see in the figure on the right-hand side, we were able to generate unique alignments for each one of these barcodes. So in this mixture of 8 different barcode samples, we were able to generate a range of alignments that uniquely identified each of the 8 barcodes, with the relative number of alignments roughly corresponding to the ratio of the peptides within the sample. And again, I think this is just highlighting how you could use these barcodes to perform selection screens where you may have that traditional genotype-phenotype linkage disrupted by the host system. So that's one example in which we've started off with a larger selection of barcodes. I now want to turn to a specific use case in which we've used the barcodes to carry out a selection screen. So in this particular example, we started off with just a simple 2 barcode system. In the first barcode, barcode 1, we've appended that to a gene encoding an MBP nanobody. And with the second barcode, barcode 2, we've appended that to a gene that encodes a GFP nanobody. We then express each one of these barcoded nanobodies, as shown here in the schematic. Those barcoded nanobodies were then functionalized with our loading complex. And then subsequently, we performed a rounds of selection using GFP-coded magnetic beads. We then underwent the traditional process of cleaving those barcodes and performing sequencing. As shown on the figure on the right-hand side, you can see we started by taking the pre-selected barcode samples, performing sequences on them prior to selection. We found that we had a ratio of barcode 2 to barcode 1 of approximately 3 prior to selection. We then performed our round of selection using the GFP immobilized onto magnetic beads and found that the ratio of the barcode 2, which again corresponds to the GFP nanobody, increased to approximately 1,000 to 1 compared to the ratio of the GFP nanobody to the MBP nanobody. So this change from approximately 3 to 1 pre-selection to approximately 1,000 to 1 post-selection represents a 300-fold enrichment of the GFP nanobody barcoded on barcode 2, and demonstrates the potential of these peptide nanobodies, enabling the ability to select and screen for novel binders or different protein engineering candidates within complex mixtures. Finally, I'd like to move on to the last use case, which is the detection of amino acid variants using next-generation protein sequencing on platinum. So in this particular use case, we have a study of a amino acid misincorporation with one of our collaborators. We began this project by taking and benchmarking the relative on-chip kinetics for a wild-type QLE peptide, the on-chip kinetics, relative coverage of each of the amino acids in this peptide are shown above. Including that, we've also demonstrated a new graph in which we have the die lifetime, which is representative of each individual recognizer in our system, versus the pulse duration, where you can clearly see how within this original wild-type peptide, we have 2 distinct populations in which we have the short pulse duration Q, shown here at the very beginning, and this longer pulse duration N within the same recognizer being identified. Once we had benchmarked the performance of this native wild-type peptide, we then introduced a series of known amino acid substitutions at different sites throughout this protein. And these mutations within the sequence included either new recognition with our current set of recognizers, or went and left the original sequence with new residues that were invisible to our current recognizer system. So this is one example of how these changes in the amino acid sequence can be detected on chip through a direct detection method. So in this particular example, we have a series of 3 different peptides. Again, we're starting at the top with the wild type benchmarked peptide with the QLE starting sequence. In the first mutation, what we've done is we've changed from this initial glutamine residue in position 1 to an asparagine in position 1. Now, this change in the amino acid sequence only results in a change of our binding kinetics and not the change in the recognition. And as you can see, while we have the same recognizer interacting with this N-terminal amino acid, we get very distinct pulse durations as recorded here on the chip, with the original glutamine residue having a pulse duration of approximately 0.3 seconds compared to the asparagine, where we now have a pulse duration of about 1.2 seconds. And this is represented here in the graph over on the right-hand side. We then decided to change this initial position 1 to a new recognizer. This is using our aromatic recognizer, and we started by taking and converting this glutamine to a tyrosine. And again, now what you have is a distinct recognizer interacting with the N-terminus, as shown here with this new purple recognition segment indicating tyrosine. And you see a corresponding change in the pulse duration with the tyrosine in the beginning. Now the pulse duration has gone from a glutamate at 0.3 seconds to tyrosine at 3.5 seconds. Again, keeping the same recognizer in position, but now just changing from a tyrosine to a phenylalanine. We now get again another corresponding change in the pulse duration, going from 3.5 seconds to a little over 10 seconds with the phenylalanine position. Again, all of the associated changes in the on-chip binding kinetics are shown here in this graph. And you can see how with platinum and its single molecule amino acid level resolution, you can detect the differences in binding kinetics, enable the recognition of multiple amino acids in this one position just by changing the recognizer and the on-chip kinetics. Now, in this is a separate case in which we've now made a mutation a little further in to the protein sequence. And what we're doing in this case is we're taking this a glutamate residue, and we're mutating it from a glutamate residue into one of a couple other different amino acids. Now, in the original wild type sequence, this glutamate is invisible, but it does influence the kinetics of the preceding residue, which is this leucine. So with leucine in this second position, just ahead of the glutamine, or the glutamate, excuse me, we have a pulse duration of approximately 0.2 seconds, which is shown here. We then make the change from this E residue into the tyrosine here, shown in this position. This adds a new recognizable residue, but it also results in a corresponding change in the pulse duration of the leucine, with going from the glutamate to the tyrosine, going from 0.29 seconds to 1.3 seconds. We can then again change this preceding residue for the leucine, or in this position 3, going from a tyrosine to a phenylalanine. That again results in a corresponding change in the pulse duration, where we were previously at 1.35 seconds with tyrosine. We're now down to just about 1 second in the presence of this phenylalanine. And then finally, what we've done is we started off with an invisible residue here with the glutamate in position 3. We've now converted this to a histidine, which again is a residue in which we do not have coverage for in our current 5 recognizer set. However, you can see that even though we're changing this is an invisible residue, it still has impacts on the ultimate on-chip pulse duration for this leucine residue, increasing the pulse duration to 0.68 seconds. So again, you can see how the power of the single molecule amino acid level resolution, how ultimately it is the combination of these sequencing effects, both the main direct recognition of the residue plus some of the downstream residues begin to influence the ultimate kinetics that are on chip, and why you don't always need 100% coverage to determine some of these residues and its impacts into the ultimate sequence. Finally, we've highlighted a few use cases where we're at the beginning of the peptide. We also want to move back down deeper into the peptide to show that we still have the same abilities. And in this particular case, now we're moving into positions 12 and 13 within this peptide. In this particular example, we've started with the native asparagine, followed by isoleucine, and we're showing, in this case, that we have a pulse duration of the asparagine at 4.15 seconds, followed by the isoleucine at 0.3 seconds. We then made a mutation in which this isoleucine has been converted to an alanine. You can see in this lifetime pulse duration plot, you now have this unique new detection of this alanine recognition, as well as a change in the pulse duration, where this longer pulse duration of the asparagine is greatly reduced down to almost half a second. And again, this just really shows you how you can take the changes in the underlying amino acid sequence level, how it influences ultimately the on-chip kinetics, and how you can use this information to identify changes in the amino acid sequence or potential post-translational modifications that may result in changes in the on-chip kinetics. So with that, I'd like to summarize. Hopefully what you've learned today is that Platinum offers a simple workflow with a convenient benchtop instrument, easy to interpret results. I think really, the power of this technology lies in its single molecule resolution that enables identifications of proteins within complex mixtures, or even within different variants within the same protein, really opening up the ability to more fully explore the complexity of the proteome. Hopefully you've also seen that the preparation of samples for sequencing is flexible and is amenable to a number of techniques, in particular immunoprecipitation. I think finally we're also introducing a few other new application areas, such as peptide barcoding. There really is a nice potential to enable selection screening where DNA-based methods linking genotype to phenotype are not always amenable. And again, just really wanted to highlight the fact that the single molecule level amino acid resolution really enables the detection of amino acid substitutions across a variety of samples. And it's really this power that would enable the ability to look for new protein variants or mutations in protein samples by taking advantage of all of the kinetic information that this system delivers. And with that, I'll take any questions. Thank you.
Thank you, Dr. Moree, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the lower left corner of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, what size proteins does platinum sequence?
So thank you for the question. Platinum is actually agnostic to protein size. There's no specific limitations in terms of the total size of the protein. We do require that proteins be digested as part of our standard workflow, so there really is no specific limitations in terms of the size of the sample.
Wonderful, thank you. Our next question here says, great technology. Do you anticipate this platform technology to be used to perform targeted proteomic applications for liquid biopsies or probe-specific protein biomarkers in tissue biopsies?
Yeah, so thank you for the question. Really great question. I think absolutely we envision this technology for these particular types of application areas. I think if you take a look back at some of the slides we presented, looking at the immunoprecipitation protocols, I think this would be a great example of that. In particular, I think for these more targeted approaches, we will require some type of enrichment, such as immunoprecipitation or maybe fractionation, that we'll be able to identify those kind of targeted samples. But I do think, this technology certainly has the ability to perform in these targeted proteomic spaces when we're looking at particular proteins of interest that may be important as a biomarker.
Excellent, thank you. Our next question that we have here says, can your technology be used to sequence the native amino terminus, e.g. immobilize an intact protein, or would you suggest to follow standard workflow and hope the amino terminal peptide is sequenced?
Yeah, again, great question. Thank you for the question. Our standard workflow really does require the ability to digest the protein of interest. We do, our library chemistry is a one-pot reaction, so all the peptides in that digestion should essentially be there and be on the chip. But yeah, so I don't know that I would say we would definitively detect the specific N-terminal amino acid, but with our technology, we do really require that digestion process to occur to prepare those peptides and immobilize them on the chip.
Thank you. This question's kind of a two-part here. It says, how long does data collection take or data analysis take?
Again, great question. So our run times are typically about 10 hours. So we collect that data over the course of a 10-hour run. It is customizable within the workflow, but we would recommend a 10 hour as kind of the standard run length. Once we complete that sequencing run, that data, if you've set it up correctly in the cloud, the data is automatically transferred and the analysis begins. Typically, I would say the overall analysis time is around 2 to 3 hours to do the complete analysis and generate the reports that you can see and visualize your data with.
Great. All right, our next question here, we have a ton of them coming in, so please, everyone, if you have questions for Dr. Moree, go ahead and type them in the Ask a Question box, and we will try to get to you today. Our next question here says, have you tested if PTMs such as glycosylation or single glycan can affect recognizer binding kinetics?
Yeah, so I think regarding PTMs, the way we kind of approach them is that with any type of modification, you would expect some change to occur within the corresponding on-chip kinetics, right? So, it may be glycosylation or phosphorylation, and what we have seen with some of in some of these cases is that you do get distinct changes to the on-chip kinetics. I think really, if you want to think about how PTM detection might work on the chip, you can look at the data that we presented toward the latter half of the presentation, looking at those kind of amino acid level resolution changes. It's really going to be based upon the detection of differences in binding kinetics that would be associated with any particular PTM.
Wonderful, thank you. Mm-hmm. All right, our next question says, could you sequence a protein with full sequence coverage, and how long will it take?
With full sequence coverage, I think right now, as we kind of highlighted during the presentation, we have currently, recognizers that recognize 11 of the potential 20 amino acids. So we have the ability to recognize directly those 11 amino acids, and then based on, as I mentioned and demonstrated during the presentation, we can indirectly infer the position or some of the downstream amino acids based upon the overall chip kinetics. Again, a sequencing run, because of our digestion and the ability to have multiple peptides that incorporate a protein of interest, we really only need about a 10-hour sequencing run to get those results.
Excellent. All right, our next question here asks, how many binding sites are available on a chip?
Yeah, so our chips, we have, it's around, I want to say, around 2 million or so apertures within the total chip. And so that is, that's essentially the number of binding sites that we might have, or the number of wells that we may have on a current chip.
Great. All right, thank you. Our next one says, can you predict the effect of downstream sequence on the binding kinetics of the detected AA?
Yes, I mean, I think we certainly can. If you think about the way that this process works, and as we kind of demonstrated again in the presentation today, the N-terminal amino acid obviously directs the great majority of the binding energy, as you would say, for the recognition event. But we do have influence, or that binding event and the on-chip kinetics are influenced by those downstream positions. And so we do utilize that information as kind of part of our algorithms to do some of the amino acid calls. And that information is really critical, I think, for really determining and allowing us to take and make our alignments and make our predictions. So we do utilize both the N-terminal amino acid and the subsequent downstream sequence to have the best understanding that we can of the binding kinetics on chip.
Great. All right. Our next question here asks, how does this technology compare to the traditional N-terminal sequence?
Oh, this is like, I would say, like Edmond degradation, right? So, I would say that in this particular case, what we're doing, I mean, we obviously have, I think our traditional kind of, you can think about it from the standpoint that we are kind of cleaving amino acids one at a time in both cases as we're trying to generate that information. We're really using, I think, the power of the single molecule assays that we have in our system, where we have these distinct set of recognizers that can unambiguously identify both the N-terminal amino acid as well as some of the downstream sequence space. The process isn't, I would say, is using like the harsh chemistries and the conditions that are undergoing in sort of the Edmond degradation process. It's also, a very straightforward technology, very simple workflows, all on like a very small kind of benchtop instrument. And so I think there are, a number of differences between those two technologies. But I think at a certain level, we are kind of going by, down peptide chains and really kind of interrogating each single amino acid in that chain. So I think that's what I would say is maybe the similarity, but the way it's done and ultimately the amount of information that you're getting from the system is going to allow, I think, a deeper understanding of that based on the chip kinetics.
Great. Okay, our next question asks, are there new reagents under development to obtain deeper sequence coverage?
I think, obviously we have, this is a very active area for us. We know that we want to enable deeper sequence coverage. And I think as we've recently announced, we do have a new set of reagents that are going to be coming out early next year that will give us a new recognizer in our system that will give us more sequence coverage. Yes.
Great. Our next question asks, what type of controls are used?
Yeah, so it's a great question. We do have, I think when we're setting up the system, we do have some control peptides that we can utilize to look at the performance of the system. I think really when you think about controls, though, you really have to kind of understand the experiment that you're doing, right? And, those are going to be kind of specific for the experiment, your actual experiments. I think one example that I would give, kind of referring back to the information that we had in terms of like the barcoding system, where we started off by taking kind of a pre-selected sample and performed our sequencing analysis on that, and then compared that to the post-selection sample. If you really want to make those kind of relative quantification estimates, you really have to set those experiments up correctly and identify the right controls. And in our particular case, it was having a pre-selection sample. And so I really think about controls as those things that are really critical for the ultimate experiment that you're running, and how they need to be set up. So.
Great. All right. Our next question here says, on some of the data you displayed, multiple stretches of amino acids were recognized or colored, but the peptide was not considered identified. Why is that?
I think I'm not. Typically, I think when we're looking at the sequence space, you're thinking about those examples of, let's say, for some of the sequence charts that we showed for something like maybe PDL1 or maybe even some of the protein complexes. So it has to do with how our peptides are being, or how the data is being analyzed. We do have, as I mentioned, a number of criteria that go into analyzing the kinetic traces, and then making sure that they have the appropriate scoring criteria that will enable the confidence that those alignments are matching to a particular individual peptide. So it's really kind of on the back end analysis side. So what we look at both, I think, is what we would anticipate the on-chip binding kinetics to look like, and as well as when we're doing our actual alignments, that data is kind of corresponding to the appropriate kinetics. And in some cases, what you'll see is that you have amino acids that are particularly to be recognized, but they may be in peptides that have, it could be very long, that maybe did not show up on our sequencing chip for some reason, such as they maybe not be, they could precipitate or something like that. Particularly if peptides are very long, we may see that to be the case. But I think really it comes down to the fact that at the end of the day, we have the ability to interrogate a number of amino acids within any peptide, but they have to have a certain alignment score that needs to be met for those peptides to be confidently identified on chip. So it's that kind of combination of the ability to see amino acids within that peptide, but then also to confidently identify them through statistical parameters that are in our algorithm.
Great. Thank you. All right, we have another question here that asks, have you used this platform for HCP analysis for QC of biopharmaceuticals?
It's a great question. I think we can certainly envision this technology for the QC of biopharmaceuticals, like biologic drugs and things of that nature. And I think, we're certainly thinking about that. I don't think we have a direct application space for that yet, or we haven't really demonstrated that in like an application note or things of that nature. But we do see there being, a nice synergy, particularly for the ability to maybe sequence antibodies or antibody drugs, because they are protein-based. And I think there's, a really great idea behind that to look at that as a validation method. So.
Excellent, thank you. All right, we have another question here. It asks, is the technology quantitative?
Again, a really great question. I would say, as we kind of demonstrated within the slides today and in this presentation, the technology is, I think, best used for relative quantification. We do report out, as we showed again throughout the presentation, we do report out like the total number of alignments and everything of that nature. But I think really, we think about this as a relative quantification at this point. I think to drive towards more absolute quantification, we need to develop like reference standards that we can utilize in the chip to help normalize values and things of that nature. But it's absolutely can be used for relative quantitative measurements at this time.
Excellent. And we have time for just one more question here today. That question asks, are people using the platform beyond peptide sequencing to monitor protein peptide or protein small molecule interactions?
Again, a really great question. Obviously, we've got people who are looking at, I think what I would call kind of more complex samples. Particularly, there's a lot of interest in the protein interaction space, or protein peptide ID space. So certainly that's, I think, a very key area of interest for this technology. And I think on the small molecule front, I think we certainly see applications in which we could look at, the use of small molecules and kind of A/B samples to detect changes in protein abundance and things of that nature upon like small molecule treatment. So I think there's really, for this technology, a lot of great space, particularly as you start thinking about these kind of, what I would say is more like the therapeutics discoveries and things of that, where this technology could really have an impact. So.
Excellent. Thank you again, Dr. Moree, for your time today. We would also like to thank LabRoots and our sponsor, Quantum Sci, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.